Oh, there we go. Great. Thanks, everybody. It's wonderful to be in person. My name is Leah Gore, and I'm a pediatric oncologist. I'm part of the Cancer Center program here. It is an absolute privilege to introduce my friend and colleague, Bona today. Dr. Bona comes to us from the Harvard Boston Children's Dana-Farber conglomerate of everything great. Kira is an assistant professor at Children's there and works specifically on health equity and in their leukemia program. Instead of kind of reviewing her CV, which anybody can do, I thought I would tell you that she is from Western Massachusetts. She has 2 dogs, 2 cats, 2 children, but only 1 husband. And she abhors everything about bananas. So your fun fact for the day, I think Kira is known as both a very gracious colleague and a gracious mentor. So I'm excited for her to meet with people afterward and talk a little bit about advancing programs. I hope some of our Cancer Center colleagues are online if they're not here in person, because I think we have a lot to learn and a lot to kind of derive from the efforts that Kira is making and will make in the future. So without further, it is a pleasure to welcome her, and I think we're going to all learn a lot.
Leah, thank you so much for that incredibly kind introduction and for my fun fact. If you guys remembered nothing other than bananas at the end of today, it will be a good day. I am so incredibly happy to be here and excited to get to share some of the research that my group is doing, but more importantly to talk a little bit about a framework for health equity research in the cancer space that I have been playing with and which I will welcome feedback and engage discussion afterward. I will start with the requisite boring things, and I may wander if that's okay. You guys just send me right back if need be. By starting to say that I have no conflicts of interest. This is a continuous source of dissatisfaction for me as a health equity investigator. I hope that in the future I will have many conflicts of interest. So if you guys have bright ideas about how I can get big pharma to make me conflicted, please let me know. I'm also supposed to make sure you guys know my CME objectives for you for today, which I am not going to read, but I hope by the end of today, perhaps you'll have an appreciation for some of the current data in pediatric oncology, thinking about exposure to adverse social determinants of health, how they're associated with inferior outcomes in childhood cancer, and how we can think about applying a relatively standard oncology framework to investigating these moving forward. My plan for the next 45 minutes or so, and my intent is to hopefully leave plenty of time for discussion, is the very straightforward one. I am going to start with some context and definition to make sure that we are all on the same page with what I'm talking about. I'm going to focus the first third of the talk on the what, presenting some smattering of data on the associations between poverty exposures and childhood cancer outcomes. And then really the bulk of what I'm going to chat about today is thinking specifically about how we can do subspecialty health equity research using pediatric oncology as a paradigmatic population. So I'm a pediatric oncologist, and that's what I'm going to talk about today. But everything that I'm speaking about today, I think is not specific to childhood cancer, but rather applies both in the medical oncology setting and in other subspecialty pediatric settings. All right, and before some actual data, I have 2 notes that I just want to share. The first is to say and to acknowledge that social determinants of health are complex and multidimensional constructs. These operate differentially at the community level, household level, at the individual level. I have a personal research interest in 2 adverse social determinants of health, income poverty and unmet basic resource needs that I am going to focus my talk on today, but to acknowledge that there are many other adverse social determinants of health that may be your favorites, and all of this I think can be applied to the investigation of those various adverse SDOH. The second thing that I am going to acknowledge today is that I am going to focus again on income poverty and unmet basic resource needs. I am not going to present data on racial and ethnic disparities in childhood cancer outcomes, nor a framework for investigating these, but I think it is necessary to note at the beginning of a talk like this that centuries of structural racism in the United States mean that children who come from historically marginalized populations are significantly more likely to be exposed to adverse social determinants of health and to therefore bear the burdens of any inferior outcome that I am talking about today. So to note that, but not going to be my focus. And then the final thing, again, today's talk focusing on childhood cancer as a paradigmatic population, but really I hope as an exemplar population for doing some of this work. All right, so let's start with the why. Why is talking about poverty and childhood cancer the most interesting, exciting, fabulous thing to discuss? I'm going to start with a story because that's what every good research talk starts with. This is a picture of Madison and the back of my much, much younger head. Maddie was one of my very first patients when I was a first-year pediatric oncology fellow in Boston. Maddie was diagnosed with garden variety B-cell acute leukemia, and she enrolled on a frontline clinical drug trial open at Dana-Farber at that time for her leukemia. Her leukemia relapsed. We successfully put it back into remission. It relapsed again. We successfully put it back into remission. But why do I tell you about Maddie? Well, I did my pediatrics residency training in Boston. My primary care clinic was at a Boston hospital called Boston Medical Center. Boston Medical Center is the safety net hospital for the city of Boston. It is the old Boston City Hospital, and it serves really an exclusively historically marginalized population of children, meaning those who are new immigrants to this country, those whose families speak languages other than English as a first language, those who are Black or brown or living in low socioeconomic backgrounds. And as a pediatrics resident in primary care clinic at BMC, I promise I'll bring this back to oncology, I had a mentor there, Dr. Debbie Frank, who's a general pediatrician, who taught me early that I was doing an abysmal job as a general pediatrician if when a 9-month-old came in for a well child visit, I failed to ask that family whether they had adequate food on the table before I sent them out the door after vaccines and assessing development. And her rationale for that was that there was 30 years of beautiful data in general pediatrics that things like food insecurity matter to a child's developmental outcome, matter to a child's health outcomes, and more importantly, that we could do something about it in the clinic that day. And that if a family screened positive for food insecurity after I gave vaccines, I could write a script for groceries. The mom would walk down the hallway to the therapeutic food pantry there and go home with a bag of groceries and come back weekly to get those. And if I failed to do that, I was actually not providing good medical care. So I did 3 years of peds residency. I absorbed that as being a statement of truth. And then I stayed in Boston to do my fellowship at Dana-Farber and Boston Children's. And I went from in June being a pediatrics resident who believed this to be true on July 1 to being a pediatric oncology fellow. And we did not ask. I learned how to do day ones and new diagnostic discussions. I learned how to provide protocolized therapy to kids with cancer. And we did not ask if you were food insecure. And we did not ask if you were income poor. And when I asked my mentors, who I revered as being some truly fantastic oncologists, why, the answer I got was a kind one, but that said, oh, well, Kira, there's disparities in general pediatrics and in early 2000s. And it turned out there were like a handful of publications showing that there were racial and ethnic disparities in outcome, but that maybe somebody should investigate socioeconomic disparities. And I wondered, maybe, is that not true? And so back to Maddie, one of my first patients, and her mom came into clinic. This was after her second relapse. And she said to me as she's breathless, 4 hours late, as was typically the case for her, you know, Kira, I was going out to the car and it wouldn't start. And it's really scary when you have an appointment an hour and a half away that's maybe life-saving to your child. And you don't know how you're going to get there. And Maddie, for me, was one of those case examples where it felt like in Gen Peds, I would do one thing and it mattered to outcome, but in oncology, we did not believe that mattered. And yet this child with garden variety B-ALL had now relapsed twice, and might there be something there? All right, that's my story going in. I promise I'll be completely data focused moving forward. So why study poverty and childhood cancer? Well, basic US epidemiology that all of you know, child poverty likely to experience trauma and have higher all-cause mortality than children not living in income poverty. And there's this little secret, which is the day before you're diagnosed with cancer, you're a general pediatrics patient. So in theory, all of those things actually apply after you're diagnosed with cancer as well. Despite that body of data in Gen Peds, poverty exposure and other adverse social determinants of health have not historically been considered risk factors in the pediatric cancer setting. And as a result, because we are very data-minded, we've never systematically collected data on these exposures in our trials or other settings, because if they don't matter, why would you bother collecting data? All right, so that's income poverty. I told you I have another research interest, which is in unmet basic resource needs. This poverty exposure is termed household material hardship. And this refers to basic resource needs all of us need on a day-to-day basis to survive, having adequate food on the table, having a roof over our head, having the heat on in the depths of winter in New England, or the air conditioning on in the South in the summer, having a car to get to a medical appointment for chemotherapy. We know from data from my group and others that 1 in 3 children, much like income poverty, lives in a home where their parent tells us they have at least one of these unmet basic needs at the time of their child's cancer diagnosis. So that means, from a practical standpoint, when I'm on service, 1 in 3 children who walk through the door with a new diagnosis of leukemia is coming from a home that is low income and is coming from a home where their family is facing at least one of these unmet basic resource needs. All right, those are the definitions, little context. I know we're not a fully pediatric audience here, since I'm using pediatric oncology as the demonstration population for these data. Simply to name the research framework we work in, childhood cancer is very rare. There are just about 16,000 children diagnosed with cancer on an annual basis across North America. As a result of this, it means that any one large center, including this one, is not going to be able to statistically power their studies by just enrolling kids in a trial at their center and figuring out is A better than B. And as a result, childhood cancer has relied very heavily on a cooperative group trial-based model of care and discovery. And this means that over the last decades, centers across North America and the world now, but we'll focus on North America, have come together to agree to all enroll children on the same clinical trials, figure out is A better than B, find a new standard of care, and then do it again every 5 years. And this model of care has been highly successful. I'm putting up here a now old curve from Steve Hunger's New England Journal paper. This is simply showing you overall survival for children with a diagnosis of acute lymphoblastic leukemia, or ALL, the most common childhood cancer, by era for kids treated on clinical trials. And the punchline of this that you're supposed to take away from it is that in the 1960s, children with a diagnosis of ALL who enrolled on a clinical trial had less than 10% overall survival. And today, children with ALL who enroll on a clinical trial have a greater than 90% overall survival. Now, I will name here for my non-liquid tumor colleagues, we have not had this success in all tumors, but this is a nice paradigmatic one, right? The trick to this, and then this is the framework for everything else I'm going to talk about, is that none of this, and conveniently for me, the curve on the top there ends in the early 2000s, none of this is new drugs. This is all old school drugs from the 1960s, the 1970s. This is dexamethasone, doxorubicin, methotrexate. And this improvement in survival was, again, not based on magical new drugs or immunotherapy, but rather on refinements in risk stratification, figuring out clinical, tumor genomic, treatment response-based risk factors for outcome, and then testing risk-adapted therapy on the basis of those risk factors and improve supportive care. And that is how we went from 10% to 90% overall survival in childhood ALL. And that has been a highly successful model in this disease, at least, one that we are replicating across other diseases as well, not with the same success. Missing from this model of discovery and care has been any consideration of social determinants of health. So hopefully I've set myself up for a pitch here of how we could then think about that a little bit differently. So what's the problem? Well, I'm going to tell you the punchline to that question right now, and then I'm going to move into showing the data. The problem is that we now know that despite that clinical trial model of discovery and care in childhood cancer, that poverty is associated with inferior outcomes across the care continuum in pediatric oncology. And I will pause here very briefly for my medical oncology colleagues to acknowledge that our problems with health equity and disparities in childhood cancer are perhaps a little bit different than in the medical oncology space, where access to highly standardized care and access to current best therapies available is a huge issue. And diversity in trial enrollment is a huge issue. And I'm just going to say, and have you perhaps access, that those are not the driving issues for disparities in childhood cancer. And so my focus is going to be on mechanisms that are really different than what are being studied in medical oncology right now, because getting kids to trial is not the biggest barrier to curing them today. All right, so what do I mean when I say that poverty is associated with poor outcome? I'm now going to walk you through a snapshot of studies to get to the important and fun part of what we're doing today, to prove the point of what I've now stated. So the first place that we began looking was in the most common childhood cancer, which is acute lymphoblastic leukemia. And the punchline here is that we found in a retrospective study that poverty is associated with early relapse in childhood ALL. What I'm showing you here is a cumulative incidence of relapse curve for children with a new diagnosis of ALL who were enrolled on a multi-center clinical drug trial for new ALL through the Dana-Farber ALL consortium over a decade. So this was 2 clinical trials. In red are children who were considered poverty exposed in this analysis. In black are children who were not poverty exposed. And I told you at the beginning of this talk that historically in childhood cancer, we haven't collected measures of poverty. So one might ask, where are these data coming from? And this is true. These decades of trials in the Dana-Farber ALL consortium had no measures of parent-reported poverty exposure or any other social determinants of health because nobody thought they were important. These trials had already been completed and published. What was available on these trials was a child's zip code at the time that they enrolled on trial. So this was a stupid, simple, quick and dirty retrospective analysis of previously published trial data, where what we did was we took the zip code at time of trial enrollment, we linked it to US Census data, and then we just dichotomized it based on a US Census definition of what a high poverty zip code is. And the United States Census says that a zip code is a high poverty zip code if more than 20% of persons in that zip code live below the federal poverty level. Very rough and dirty metric, very prone to misclassification bias. In this analysis, what you see there is that those curves ultimately overlapped. There was not a difference in absolute relapse between the two cohorts. What was different was how quickly they got there. And you can see that the red line gets there a whole heck of a lot quicker. And the primary finding from this study was that children who were poverty exposed, here proxied at the area zip code level, were significantly more likely to experience early relapse, defined as less than 36 months in complete remission. And as a clinician, this was important because we know in acute lymphoblastic leukemia that time to relapse is one of the strongest predictors of our ability to successfully put that leukemia into a second remission. So this felt important to me as an early career investigator, but the feedback I got was, well, that's ALL, most of it's outpatient, probably it's just there, it's about adherence and access to care. It's not gonna hold true across all the rest of childhood cancer. So the next place we looked was in a solid tumor that is very different, and that is pediatric neuroblastoma. Neuroblastoma is the most common solid tumor in children, and neuroblastoma, high-risk neuroblastoma in particular, is treated very differently than ALL with intensive intravenous inpatient chemotherapy. Here again, you'll note a similarity in methodology. We took previously published data from a clinical trial here run by the Children's Oncology Group at over 100 centers. This trial had been examining the addition of dinutuximab, which is a targeted immunotherapy agent to a standard chemotherapy backbone. And trial was actually stopped early because it turned out dinutuximab had the greatest improvement in event-free survival that we'd seen in decades for kids with high-risk neuroblastoma. So it seemed like a great place to look. So we took data from that trial, just kids who got dinutuximab, and we dichotomized them based on proxied poverty exposure. Here again, COG had not collected any data on family-reported social determinants of health. We had access to a child's insurance status at the time they enrolled on trial. Again, rough and dirty. We dichotomized that, saying that children who were publicly insured with Medicaid or CHIP, we would consider to be exposed to low household income. And that's because the majority of children in the United States who are on Medicaid or CHIP insurance come from a low household income house, and that's how they qualify for Medicaid. The punchline here, there's a Kaplan-Meier curve of event-free survival for children treated on these two trials, AMBL 0032 and 0931. In red are children who were not exposed to proxy poverty. In black are children who were exposed to proxy poverty. And the punchline is that there was a 25% difference in event-free survival between poverty-exposed and unexposed children treated on this trial across COG institutions. And then when we regressed in Cox regression models using all the previously published tumor genomic and other risk factors for poor outcome, it turned out that proxy poverty exposure was independently associated with event-free survival with a hazard ratio of just about 2. And that's an order of magnitude that is what we look for when we have a drug that works really, really well for improving things, and suggested that perhaps disparities in trial-based outcome are not limited only to diseases like ALL, treated primarily in the outpatient setting, but also to those diseases that we treat primarily in the inpatient setting. Poverty is not only associated with relapse and survival, it is also associated with important patient-reported outcomes, including parent and child distress. Here I'm showing you data from an investigation led by a fantastic early career investigator, Dr. Pooja Umaratilla, who's an assistant professor at UT Southwestern. And Dr. Umaratilla took data from a frontline trial for children with acute lymphoblastic leukemia. Here we did have access to parent-reported data on poverty exposures, specifically to that measure household material hardship I told you about before, food, housing, utility, and security. And she asked the question, does parent distress vary on the basis of exposure to household material hardship? This is a bar graph showing parental reports of severe psychological distress using a measure called the Kessler-6. And the punchline of this study is that at the time of a child's diagnosis and enrollment on trial, poverty-exposed parents were twice as likely to experience severe psychological distress. And 6 months into chemotherapy, despite the same intensive social work engagement, poverty-exposed parents are 5 times as likely to experience severe psychological distress when compared to unexposed parents. And you might say, well, we're not in the business of treating parental distress in childhood cancer, and there is some truth to that, although we could say that maybe we care about it. But in ALL, which is a disease we primarily treat in the outpatient setting, where we ask parents to give oral chemotherapy at home for 2 years, you might think that perhaps parental psychological distress could impact how well they're able to adhere to therapy. But it's not just parents. These are data from an analysis led by another fantastic early career investigator, Dr. Maya Ilowite, who's at Dana-Farber and Boston Children's. Dr. Ilowite took data from a multi-center palliative care intervention trial and asked the question, were children with advanced cancer who lived in low-income homes, here self-reported by parents, having the same symptom burden as children not from low-income homes? In blue are children who lived in low-income homes. In orange are children who did not. And the punchline of this analysis was, among a cohort of children with advanced cancer enrolled on a palliative care intervention trial at 3 major academic pediatric oncology centers, low-income children were significantly more likely to experience pain as compared to non-low-income children, despite what was considered systematic care delivery. Okay. That's a smattering of data, and I'm going to tell you the punchline I took away from that, leading then into the work that my group is doing today. You don't have to believe it, and you can push back on it at the end of the talk, but my punchline was, perhaps poverty could be an intervention target. And the rationale there is, if despite standardized care on clinical trials in a multi-center fashion, children living in poverty are more likely to relapse and die, and children and parents are more likely to experience distress, then the takeaway for me is that ensuring access to clinical trials in childhood cancer is simply inadequate if our goal is to cure all children of cancer, or if our goal is to ensure equal patient-reported and quality of life outcomes, which of course begs the most important question, how do we intervene to improve outcomes? And I'm going to put here the next slide that is usually the question and or the comment that everybody says, which is, so what? Disparities are not new. This is absolutely true. Just because I showed you a couple years of pediatric oncology studies that I did that are rough and dirty and retrospective, everybody knows disparities exist. And despite that, the needle has really not moved in the last half century. So why would we think that it could be different in an oncology or a subspecialty pediatric setting? Well, I would say I think there's actually an enormous opportunity in pediatric oncology in particular, but in subspecialties in general, to leverage a clinical trial model of investigation to fundamentally change the approach that we take to disparities research. So what do I mean by that? What if we were to consider poverty to be a druggable target? What if we were to take that frame that we as oncologists are very comfortable thinking about when we think about our next trials or how we're going to next improve outcome, and apply that same concept to poverty or to other adverse social determinants of health? What could that look like? I'm bringing you back here to the prior approach that we have applied in pediatric oncology here, using ALL as an example that's been highly successful. What if we were to actively integrate social determinants of health into this framework of investigation? Well, please bear with me for one more little analogy, and then we'll move into current data. What if we thought about social determinants as being the equivalent of cancer genetics? I'm putting back up here that Kaplan-Meier curve of event-free survival for children with high-risk neuroblastoma. And I will tell you that when I first presented these data to colleagues in Boston, one point of feedback I got was, Kira, those are really stunning data. And that is incredibly depressing, because what in the world are you going to do about poverty in a clinical trial run through the Children's Oncology Group? But if I walked into a conference room of pediatric oncologists and I changed this slide and I said, guys, I just reanalyzed data from ANBL 0032 and 0931, and guess what? Here I annotate it. I just found that this gene mutation in neuroblastoma samples is associated with a 25% decrement in event-free survival. There's nobody in that room who would say, oh, that is depressing. What are you going to do about it? In fact, people are enthused about that. This is literally the bread and butter of what we do in oncology, right? We go back to our trials, we identify new tumor genomic or response-based risk criteria for relapse or treatment resistance, and then we literally have a recipe for what we do moving forward. And this recipe is not exactly rocket science, and forgive me for breaking this down and making it sound more stupid simple than it is, but it's pretty straightforward, right? We identify the new gene mutation associated with relapse in the last trial, and then moving forward, we do 3 things. The first is we prospectively collect data on that gene mutation in the next trial to make sure we got it right. The second thing is that we ask our lab-based investigators to begin sussing out the mechanisms driving treatment resistance associated with that gene mutation. And the third, and this from my perspective is the most important, we drug it. And we don't wait to drug it until we have figured out the mechanism underlying that excess risk of relapse. We drug it in the next trial. We consider it to be a high-risk feature, and we intensify the therapy that we're giving you. So we first start by intensifying cytotoxic chemotherapy. If it's neuroblastoma, maybe we give you another transplant. And then eventually, when our lab-based investigators figure out the mechanism, then we introduce targeted therapies. But we don't wait to drug it until 20 years later we know why, because it's not ethical to let kids who we know have a risk factor die while we're figuring that out. And I think that perhaps we could take the same recipe and apply it to social determinants of health, treating them just like cancer gene mutations. allow me to take a sip of water, and then I will keep going. And so this framework is the approach that my research group is currently taking, and that I'm going to pitch as being one that we could apply in a couple different settings. And that is that following that recipe, we are currently thinking about trial-embedded prospective collection of social determinants of health data, correlative biology and S2H investigation, thinking about mechanisms driving treatment resistance and excess relapse, and trial-embedded health equity interventions. And I'm going to walk you through some of the work that we're doing now that hits on each of these really very basic pieces of a recipe, and then talk a little bit about where we're going moving forward. And I'll find a clock to make sure I'm not over time as well. And also, it's so nice to be in person. Please interrupt me if you have questions. A dialogue or discussion is much more enjoyable than my talking at you as I keep going here. All right, so starting with really the most stupid simple, which is prospective collection of the data. If this were a gene mutation, we know how we would do that, right? In the next trial, we would have standardized biospecimen collections where we're going to be looking at that prospectively in different labs along the way. What does that mean when you're talking about SUH? So we are currently leading some of the first US trial-embedded studies of poverty exposure in childhood cancer in 2 different consortia. The first in the Dana-Farber ALL consortium, which was purely because I work at Dana-Farber, and as an early career investigator, I had access to a PI who was willing to let me try this when nobody had done it before. And it's really nice to leverage the community that you have near you and have them be willing to collaborate. And then eventually we've expanded this now to the Children's Oncology group. And the concept here is that if we are collecting data at the time of child's trial enrollment, we will then have those social determinants of health data available to annotate trial-collected clinical outcomes, such as relapse, survival, toxicity, as well as to annotate biospecimens, which have been collected across the course of that trial, to hopefully in the future be thinking about biological mechanisms driving disparate outcomes. I'm going to tell you how this is working, and I can say that over the last 5 years, we now very confidently in the pediatric cooperative group setting can say that this approach to trial-embedded collection of parent-reported social determinants of health is feasible, it's acceptable, and not shockingly, poverty is highly prevalent, which we already knew. What do I mean when I say that? And here I'm going to show you data from the Dana-Farber ALL consortium, but I can tell you they're nearly identical in the COG. From a feasible perspective, we do this as trial-embedded opt-in survey data collection, which means that at the time of a parent's consent to clinical trial for their child, in the same way that they're offered an opportunity to opt in to biospecimen banking, they're also offered an opportunity to opt in to completing a brief SUH survey. In the Dana-Farber ALL consortium and now across COG, I can say that with this approach, 88% of parents opt in to complete SUH surveys. It's acceptable from the perspective that once they opt in, because many people at the beginning said to me, oh, they'll say yes because nobody knows what they're talking about when they consent to a clinical trial. But then when you actually come to them with a survey, they're going to say, no, I actually didn't sign up for that. I don't want to do it. Or no, this is too sensitive to collect the data. Turns out that's nonsense. Among those who opt in, we have 96% complete survey data at a baseline time point. And with that, I told you my primary exposure of interest is household material hardship. We have 99% complete HMH data. This is true in the Dana-Farber ALL consortium. This is true in the Children's Oncology group. Parents are happy to participate in research on this topic. They're happy to complete surveys, and they do not find these topics too sensitive to share the data with us. And then poverty is highly prevalent. In the Farber consortium, at the time of diagnosis, this is across children with a new diagnosis of ALL at 8 US and Canadian centers, 34% of children at time of trial enrollment live in a home where their parent tells us they have at least one food, housing, or utility insecurity. We've now done this in the COG as well. We've collected over 1,500 baseline SUH surveys, which is the largest SUH data set to exist in childhood cancer. And that's across 194 contributing sites and in greater than 9 languages. So the other important feasibility takeaway for this is it does not require a local champion to make it happen. This can occur in the cooperative group setting at centers all across North America, and it is not limited to families who speak English. We can do this across all of our patients and families who are participating in our trials. Not to perseverate on this, moving on to the other buckets of investigation. So you have to collect the data, and then I said, then we think about lab-based investigators doing translational research to figure out mechanisms. And this is the second approach that we're taking, and that is to begin thinking about social determinants of health and correlative biology here in collaboration with colleagues who run wet labs and actually do wet lab research. And so from this perspective, just to give you the frame that I and my group have been thinking of, coming into this, one of the big questions I have increasingly had is, are disparities actually all driven by socio-behavioral mechanisms? And what do I mean when I say that? So In the disparity space, it is incredibly common, whether you're talking in cancer or in general pediatrics, to talk about very standard sociobehavioral mechanisms of access to care. Are you able to get where you need to be to receive the care that you need? And adherence to recommended treatment. And these are things that have been published across many diseases and are known to be associated with inferior outcome. But particularly going back to those neuroblastoma data, where most of those children were inpatient, receiving intravenous chemotherapy, I really wonder whether all of that can be explained by access to care and adherence to recommended treatment. And that for me begs the question, is it possible that there is underlying biology associated with social determinants of health that could explain some of the differential outcomes that we're observing? And why might that be? And so here we're really applying basic general pediatrics concepts. So the idea of toxic stress, which is a concept derived originally in general pediatrics and refers to a relatively basic concept, which is that if you take a child and you chronically expose that child to stress, which could be living in poverty, but also could be living in a war-torn environment or being exposed to abuse, that all of us have physiologic responses to stress. And that physiologic response is that we upregulate our HPA axis, we have downstream cascade of stress hormones, and there are downstream tissue responses to those stress hormones. In the cancer space, forget Gen Peds for a moment, we talk and think a lot about epigenetics, how genes are turned on and off by our environment, where we don't usually think about stress, but stress is something in our environment. If children are developing cancer in the setting of chronic exposure to toxic stress, is it possible that this might result in treatment resistance? And so this is a kind of a question and framework that I've been investigating with a number of colleagues, three of them up here, Dr. Birgit Knokel, Dr. Jana Pickman, and Dr. Kim Stegmeyer, to begin thinking about this specifically in the ALL space, but then hopefully moving forward across other diseases. And to give you just a quick snapshot of some work we're doing in this space that's still exploratory at this point in time, Dr. Knokel and I did an exploratory analysis taking data from children with a new diagnosis of acute lymphoblastic leukemia who had banked biospecimens from the time of diagnosis. We took B lymphoblast at the time of their diagnostic bone marrow, and then I annotated those lymphoblasts as either being poverty exposed or not poverty exposed based on parent-reported social determinants of health. And then in her lab, Birgit did single-cell RNA and ATAC-seq of those diagnostic biospecimens. And the punchline, again, small exploratory investigation, is that when looking between the poverty-exposed and unexposed lymphoblast, that there's altered gene expression in pathways of glucocorticoid signaling, hypoxia, glycolysis, and fatty acid metabolism, pathways that have been implicated in treatment resistance in other cancers, and certainly also in the leukemia space. There's also open chromatin near these genes, suggesting that there's altered enhancer gene regulation. This exploratory study is not definitive, maybe it's not going to be replicated, but it suggests that perhaps there are biological responses to children who are exposed to poverty in their lymphoblasts at the time of diagnosis. And there's an opportunity to think about this moving forward. So we're currently thinking kind of in this space across a number of diseases and from a number of perspectives. And I've just put up here a smattering of projects to say that it doesn't have to just be thinking about poverty and stress exposure at the time of diagnosis and treatment resistance. We can also be thinking across the cancer continuum with regard to thinking about late effects, for example, or thinking about upfront prognostication. So as examples, in acute lymphoblastic leukemia, neurocognitive late effects are one of the biggest late effects that our survivors and their families worry about. And we know that children who live in poverty are more likely to experience neurocognitive late effects in survivorship after childhood ALL. So with Peter Cole, I'm looking at the question of whether poverty-associated CNS oxidative stress might be associated with declines in neurocognitive function during therapy. And this is in the context of the current frontline COG low-risk ALL trial. In neuroblastoma, I showed you data on excess relapse among children with high-risk disease who are poverty exposed. And so with Kelly Goldsmith, we're looking at the question of whether tumor and peripheral immune signatures vary between poverty exposed and unexposed children at the time of their high-risk neuroblastoma diagnosis. And then Dr. Haley Newman, who's a fantastic early career investigator at CHOP, is asking questions about how we should be interpreting genomic prognosticators as we think about bringing them up front in trials. So when we identify a new mutation that is associated with an excess risk of relapse, should we be thinking, for example, about the ancestral background of the host in which that disease and gene mutation was identified, and might that impact the prognostic value of that mutation? All right, so that's collaborative translational research. And I'm going to shift gears for the end here to think about the really fun part of what do you do about it? So how could we think about health equity interventions here in a pediatric oncology space? And I'm going to walk you through the framework that I have been using right now to think about intervention development. And again, we'll very much welcome thoughts and other iterations on this. And this framework is pretty straightforward and really comes back to the basics, which is that historically, in pediatric oncology, we have not had any poverty-focused health equity interventions. They simply don't exist. So when we say, oh, we've identified this in the trial, what are we going to do to intervene on it? There is not a portfolio of interventions to pick and choose from to test in the next trial. So my first premise is I think we need to start somewhere. And the rationale for this, oftentimes, again, going back to the feedback from those Kaplan-Meier curves I showed you annotated with poverty and not poverty, is that this is too big. You can't fix poverty. You're not going to cure poverty. Clearly, you can't actually fix it in a clinical trial. And again, I think that's kind of nonsense, particularly in pediatric oncology. We do first-in-child drugs. We give kids molecules that have never been put in small little bodies before, and we do it to figure out how to cure cancer. I actually don't think there's any risk or barrier to doing a social equity intervention in the childhood cancer space, and I think we're really well positioned to think about first-in-trial social equity interventions and how they impact outcome. I think the second framework we have to think about in pediatric oncology in particular is borrowing from what we've learned already in the drug space, and that is the very basic concept of multi-agent chemotherapy. In childhood cancer, we know that one drug does not cure all cancer. Very similarly, there is not going to be one health equity intervention that cures all disparities in childhood cancer. And more importantly, most health equity interventions are going to fail, just like most drugs fail when we bring them forward and we test them. And that's okay. What we need is a portfolio of interventions that are varying the social target, the delivery mode, the dose, the duration, in the same way that we think about drugs in this setting. The third, and this for me is just figuring out how I engage with colleagues who do not do health equity research, is I think we need to choose common language as we're developing interventions, common language that means similar things. Phase 1, phase 2, phase 3. Now, clearly, when we're talking about drugs, phase 1 and phase 2 have very specific meanings, but I think we can borrow that framework to talk about pediatric health equity interventions. For me, phase 1 is pilot refinement. We come up with an idea, we give that intervention to families, we do mixed methods research, we get interviews from them that tell us what's stupid about the intervention, what works well, what needs to be refined. And at the end of that, you have a refined healthcare delivery intervention. For us, phase 2 is randomized feasibility signal finding, very similar to drugs in some ways. And then phase 3 is a classic efficacy trial. All right, and then the final thing in pediatric oncology in particular, and I think this does not necessarily need to be the same in medical oncology, but due to our numbers, there has to be an eye toward scalability. And so what do I mean when I say that? I said at the beginning, 16,000 kids are diagnosed with cancer annually across North America. That means that if I develop an intervention in Boston that is very good at addressing health equity issues in Boston, that is going to do literally nothing to improve health outcomes in childhood cancer if it cannot be scaled to Denver or to Birmingham or to some small town in New Mexico. Because the reality is our catchment area for childhood cancer is the entire North American continent. And so we need things that we can be testing across populations across all of the country. All right, with all of that rambling, let me walk you through now some of the work that we're doing here. So the first intervention that we developed is called PediCare, which stands for Pediatric Cancer Resource Equity. PediCare's target is household material hardship, food, housing, utility, and security. And it includes two components. One is the standardized provision of groceries in a dollar amount that is standardized based on household size, mocked up off of food stamp benefits, and delivered via an online platform called Instacart that some of you may have heard of. Instacart was chosen not because I have any attachment to Instacart, but because from an eye toward scalability, Instacart is available currently in 85% of US zip codes. That allows a lot of opportunity for scale when you're thinking about trying to evaluate an intervention across multiple settings. Standardized gas cards based on distance from the hospital. We developed this intervention in Boston. We piloted it and refined it in a phase 1 study in Boston, where we gave it to families. We did interviews to find out what worked, what didn't work, and then we refined it. And then once we refined it, we moved on to doing what we called our randomized pilot feasibility or phase 2 study. This was conducted at 2 centers, Dana-Farber Cancer Institute, because that's where I work, and University of Alabama, Birmingham in Birmingham, Alabama, with the idea being that if we were going to be evaluating the feasibility of an intervention, I wanted 2 very different patient populations two very different centers, two very different cities in the United States, and Birmingham and Boston are not identical. We enrolled 40 families of children with a new diagnosis of cancer who had screened positive for household material hardship exposure. And those families were enrolled and randomized 1-to-1 by site to receive either the pediatric intervention over 6 months or to receive usual supportive care. And the endpoint of this randomized feasibility study was very basic. We defined feasibility as greater than 75% consent to randomization and less than 20% attrition. And the rationale for this was, I didn't know if families would be willing to be randomized to a health equity intervention like this. In fact, I was told that families would not be willing to be randomized to a health equity intervention like this. And if families were not willing to be randomized, then that means you cannot move on to then a standardized phase 3 randomized controlled trial. You have to come up with some different design in terms of thinking about efficacy. So we ran this over the course, it was supposed to be a year. Pandemic intervened. It took a little bit longer than a year when clinical research shut down. And the punchline of this study was that among 40 families, we had a 100% consent to participate and to be randomized. We had 0% attrition in either arm over 6 months, and we had 100% successful intervention delivery to the half of the cohort randomized to intervention. And we took these data to say, it's feasible to do this. I'll admit right here, feasible in a tiny 2-center randomized pilot feasibility study. But on basis of that, sometimes you have to go big or go home. So our next step is to do what I am tongue-in-cheek calling a first-in-class drug trial embedded health equity intervention. And so in the next Dana-Farber ALL consortium drug trial, which will enroll newly diagnosed ALL across 8 US and Canadian centers, the pediatric intervention will be embedded as an opt-in intervention with families randomized to receipt of pediatric or usual supportive care over 6 months as an embedded component of the drug trial. I showed you data before on ALL. We're similarly interested in thinking about neuroblastoma, where we have similarly well-published data now in terms of disparities. Here, we've not yet successfully figured out how to do this as a COG trial embedded study for many reasons I could wax poetic about at the end of this, but I will not right now. And so instead, we're trying a workaround methodology of what we call a companion trial. And this is a standalone randomized health equity trial of the pediatric intervention, for which we will co-enroll children who are newly diagnosed with high-risk neuroblastoma and enrolled on the Children's Oncology Group High-Risk Neuroblastoma Trial 2131. All right, that's a first health equity intervention. I'm going to tell you about one more, and then I'm going to wrap up to shift gears talking about where I think we need to go from here, and then hopefully end with time for questions and comments. So Pediatric targets basic resource needs by direct provision of groceries and transportation. I think that may be helpful. I don't know yet. Probably it will be a null result trial because that's usually what happens the first couple times that you intervene. And one of the big problems with direct resource provision is it does not address all of the unmet needs that many families have. And so if you shift instead to thinking about income poverty, for example, giving you groceries and transportation can allow you to reallocate available cash resources, for example, to pay the rent, because you're no longer paying for groceries, but maybe not enough to pay the rent. And that was feedback we got qualitatively from families. So Pediatric Rise is our next intervention. And Pediatric Rise has a different target, and this target is income poverty. And it includes 2 components. The first is an opt-in meeting with a certified benefits counselor. And the second, this is the core piece of the intervention, is an unrestricted cash transfer, which is just what it sounds like. This is providing cash to families with no strings attached. Families receive cash in a standardized dollar amount based on child dependence. This is mocked up off of something called the child tax credit that many of you probably heard about, where families who had children in the United States got a check from the federal government on the basis of the number of dependents in the household. That population-based intervention had one of the greatest reductions in child poverty in the United States that's happened in a very long time. So similar concept here. And families receive that cash delivered to them twice per month for 6 months. And the cash is delivered by a partnering nonprofit organization called the Fund for Guaranteed Income. There are multiple reasons for that, again, that I can get into at the end if that is helpful. This is a centrally delivered intervention, and the concept being here that you can, in theory, centrally enroll and centrally randomize and deliver the intervention to families. We have just finished pilot refining this in Boston, and we'll be activating a randomized phase 2 feasibility study in Boston and at Columbia University in New York City. I hope at the end of this month, with the goal of having feasibility data on whether we can successfully conduct this across 2 centers in a randomized fashion in the next year. So that's some of the ongoing work right now. But the big question, of course, is, all right, that's one group. It's a very small handful of studies, none of which are actually going to change the world. Where do we go next when we are thinking about health equity research in a subspecialty space like childhood cancer? And for me, I think there's really the big question of what it's going to take to effectively intervene on disparities here again in childhood cancer as that demonstration population. And I think there are a couple key things. The first is the narrative. And I think in my field of pediatric hematology oncology, but also in other fields, we need to be pushing and accepting a narrative that has permission to fail. And again, here I will come back to that example of drugs. In childhood cancer, we're very good at testing different drugs in different combinations, in different trials, and a lot of them fail. So I gave you an example of ALL where we honestly have had wild success over the last 40 years, but we could put up there instead curves for osteosarcoma, where our last three phase 3 trials were abject failures. We are not improving our outcomes in that disease. And there is nobody in the field of pediatric oncology you could say, oh, guys, we failed 3 times, should we stop? We'll just let those children relapse and die. Of course not. That's not the approach you take. It's okay to try and try again and reinvent it and come up with new targets. And we're gonna have to have that same approach in health equity, because when I fail with PediCare and I fail with pediatric RISE, and the next 3, that has to be okay until we find the drug equivalent that works. I think the second thing we're gonna need is alignment with cooperative group trial infrastructure. And this really, I think, for me, falls in 3 big buckets. I think we need to codify the relevance of this research with the concept of established cooperative group roles. We need to be embedding people in clinical trial development and delivery to make sure these questions are being asked from the get-go. And then eventually we will need NCI collaboration, certainly in the cancer space. In other subspecialty spaces, we'll need the relevant NIH partners and other foundation partners for that. And then the third, and this is the last thing that I'll end on, is I think we're going to need an establishment of an intervention infrastructure. I said we don't have a portfolio of health equity interventions to play with to see what works. We have to develop that. And actually that means we have to develop 3 things. We need a portfolio of interventions. We need a pipeline for the evaluation of that portfolio, for doing pilot refinement, for doing randomized feasibility testing. And then we need an early career training pipeline, because I'm already middle-aged in mid-career and I'm going to be outsourced soon. And we need a cadre of investigators who are wildly excited about coming up with this pipeline, just like Big Pharma, who comes up with drugs, but they don't pay you as much for this. So for that, in terms of what's going on now and where I think we can go from the portfolio perspective, I'm excited about this. I've put up here 3 examples of health equity interventions currently in development, and just giving some face time to some fantastic early career investigators. Dr. Rahel Aziz-Bose is focused on health equity in survivorship from childhood cancer. She is currently pilot refining an intervention called CHEF, which is focused on food insecurity and cardiovascular late effects. Dr. Pooja Umaratia is interested in disparities in advanced cancer and thinking about what happens to our children who are experiencing relapse and progression. And she's currently piloting and refining an intervention called ASSIST, which provides centralized benefits screening and assisted enrollment. And then Dr. Colleen Kelly is focused in the hematologic space in sickle cell disease, thinking about pilot refining an intervention called ACCESS, to how we will provide access to children who are going to be receiving high resource, high intensity, centralized interventions like gene therapy for sickle cell disease, or like CAR T therapy in oncology, or like MIBG in oncology, with the idea being that if we can break that nut and figure out how to give people access, you can improve things down the line. From a evaluation pipeline perspective, I'm excited that we're going to be launching a new health equity research consortium called IGNITE, which is a consortium of like-minded health equity investigators across the country who are willing to come together and think about how we can collaborate to more rapidly do the science of advancing health equity research, serving as sites for one another's investigation and protocols. You guys here will be participating in this consortium, which is as yet unfunded, but will be funded soon as we figure that out. And then the final piece to say the training pipeline, this is something where I can very positively say, I think there is a huge cadre of early career investigators who are excited about good science in this space, whether mechanistic translational biology, whether health equity intervention development, whether trial embedded descriptive work. And I think the opportunities moving forward are really exciting. I will end by saying, I think we're at an inflection point, certainly in pediatric cancer, but in many of the subspecialties, where we have a concurrent opportunity to both reduce disparities and improve outcomes. And that all starts from a very basic premise, which is that if we can cure childhood cancer, which we can, by the way, we can cure poverty-associated disparities in cancer. And I think the next steps are really thinking about poverty or other social determinants of health as independent risk factors for outcome, which can be drugged just like any other risk factor. And then we can apply a framework that we've used in cancer now for decades to begin investigating that in a really bite-sized, protocolized fashion, which is data collection in every population and trial, intervention, development, and testing, just like new drugs, and translational science. I will end with one of my very favorite quotes from Angela Davis, which says simply that you have to act as if it were possible to radically transform the world, and you have to do it all the time. I think this applies to any scientific investigation, certainly to health equity, but not limited to that. And I think is a good way to think about advancing outcomes for children with cancer as we move forward. I want to acknowledge the patients and families who contributed their time and data to all of the curves and other data that I showed you today, my fantastic collaborators and my awesome lab and the funders who made it possible. And I believe I am supposed to let you know that you can scan this and give an evaluation of today's talk, and I'm happy to take any questions. Thank you guys for your time.
Two-part question. One, did you see a difference in, or do we have the data to see if there's a difference in survival during that time period of reduced childhood poverty? And also, is there a time component of how long that child is in poverty? Because, dynamics change, or is that chopping up the data too far?
Those are two fantastic questions. The first one, which is a simple answer, those data are not mature. So the child tax credit happened in the midst of trials that were actively accruing or do not yet have mature survival data. Yeah. Whether we're going to successfully be able to look at trends given small numbers, I don't know, but there are plans to look at that. Yeah. The second one, I think, is a fascinating question to which I do not know the answer. I don't think anybody knows the answer. But this question of, does it matter how long you are exposed to an adverse social determinant of health, be that income poverty or something else? And I don't think anybody knows the answer. There are some interesting data in the internal medicine, general pediatric space, thinking broadly about concepts of allostatic load and wear and tear on the body. And if you're chronically exposed to something that is driving comorbidities, that your outcomes may be worse than if it's for a very short, discrete period of time where you may not have that downstream effect on your tissues and body. But I think in pediatrics, we don't yet know the answer to that. There's a very interesting focus, and then I will be quiet because you didn't want this long an answer. A very interesting focus in early child development in pediatrics, nothing to do with cancer, where this big focus, for example, on neurodevelopment and windows of time when you have the most rapid brain development, and therefore exposure to poverty may have much bigger impact on downstream core outcomes than, for example, if you take that very same child and expose them to the very same adverse social determinants of health, but later on in development that maybe the impact is not as long. So probably in pediatrics, it's going to be an age, by duration, by depth of exposure, and I think we just don't know yet.
Thank you.
Meg?
Great. My question for you, as someone who does the drug development and struggles with funding and industry sponsorship, all of your interventions require dollars. You've been great in getting funding, but to scale it up, I mean, what is your thoughts, or are there plans, or do we really need to think outside the box about how we fund these? Because if we're giving cash donations or cash distributions to families twice a month, where is that money ultimately going to come from? We struggle in Peach to get that development as is. What sort of approaches are you looking at that, or is that still a big challenge?
I'm so glad you asked the question. So the answer for right now is we're testing it, so it's easy. You fund it the same way you fund, well, drugs, I guess sometimes you fund from pharma. You fund this with foundation grants, NIH grants, philanthropy. The long-term question, and this is the forever question in this space, is how do you sustain it? Because we can't pay for things like giving cash to families. I will push back on that and then tell you how I think we prove it. And actually, I have no idea because I haven't done this yet. I also think that's nonsense. So again, these interventions may not work, but if they do, the average cost of one of the interventions I showed you is about $10,000 per child. The average cost of most drugs that you're going to give children is far more than $10,000 per child. I think in theory, one could design a very nice outcome study where, for example, you're looking at cost effectiveness and you pick the stakeholder of interest, perhaps it's an insurance company. I may be interested in relapse and survival, but if it turns out that providing groceries or cash to families makes you stay in-house 3 days shorter when you are getting your chemotherapy, the cost of staying at a quaternary care pediatric center for 3 days extra is actually quite a bit of money. And in fact, it may be a lot cheaper to provide some upfront supportive care resources to them. So I do think there's a cost effectiveness argument that can be made with stakeholders like insurance companies. There's some interesting policy perspectives, which I did not talk about today, but I think will be opportunities moving forward. And then the real answer is, I don't know, but I think first we have to find things that are efficacious. And then once we do that, we figure out how to sustain them. Yes.
And my understanding of several of these studies so far is they're looking at childhood poverty and material hardship status at diagnosis. Is there any data on how the diagnosis and how that may change a family's status if they were living kind of just above the poverty line, but then a parent had to quit their job or whatnot, how that affects a child's outcome or how these interventions may play out or not yet?
That is a fantastic question. The common term in the literature for what you're talking about is financial toxicity, although there's currently a lot of debate about whether that term should be carried forward. Anyway, that's what you would find in the published literature. And that is the concept of if we take you and we make your child get intensive therapy for a prolonged period of time, we disrupt work, we disrupt income flows, and families fall into poverty, even if they did not start there. We know from some data in general pediatrics that about 10 to 15% of families who were above the federal poverty level will fall below the federal poverty level during their child's cancer treatment. We also know that other metrics of poverty exposure, like household material hardship, increase over time. So at diagnosis, across trials. And so eventually, once those trials are mature, we can ask, does it matter at diagnosis? Does it matter at the end of 2 years of therapy? And what does it matter for, whether you're talking about relapse, survival, toxicity, late effects? So I think in the future, there will be great opportunities.
Hi, I was just more curious about the food part. So it's great that you're giving families budgets for food, but is there any programs for to educate parents about nutrition and any way that you can ensure with the money that you are giving them that the parents are able to provide healthy nutritional food for their children?
That is a fantastic question. None of the work that I or my group are doing right now is focused on nutrition education or that piece. There's quite a nice body of literature, particularly in general pediatrics, thinking about combined nutrition and food as medicine interventions in Gen Peds to improve outcomes, many of which have actually been demonstrated to have positive impact on outcomes, for example, with regard to cardiovascular morbidity in general pediatrics, but not yet in pediatric oncology. I will tell you, as a bias, I am starting from the perspective that first we just need to meet your basic needs, and you need to not be worried about you or your child being hungry, and then we'll deal with the refined nuances of whether that includes a nice leafy green along with other things, which should be there. I'm not making light of that, but I feel like we're starting at such a basic level that until we've figured that piece out of how we can systematically deliver it, it'll take a little more complexity, but absolutely needs to be there longer term. And if anybody's doing that, I'd love to collaborate with you.
The online question says, how can you justify, oops, let me go up above one more. You have already identified a gene in one of the diseases, such as glucocorticoid. How would you change the intervention?
Referring back to the exploratory data in diagnostic B lymphoblasts and showing differential gene expression and pathways of glucocorticoid. I'm assuming that's what that refers to. I think the question is, what do you do about it if you ultimately find different pathways that are up or down regulated there? And the long-term answer to that would be, if we systematically find, for example, that children who are poverty exposed are more likely to have glucocorticoid resistance, well, I think probably there's some basic changes you make there. In the same way that when we have children come to us with a new diagnosis of ALL, who've been treated for a rheumatologic disease with steroids for 6 months, we choose to use steroid-sparing regimens. We change the chemotherapy backbone that we use to choose drugs that we think will actually induce remission. And so I think eventually a similar thing can happen in this space. That cannot happen on the basis of an N=14 exploratory study, but if we replicate that multiple times, then I think that's where we'd go.
There's one more question online and a couple in the audience. Let's do
Yes.
Katie.
So one of the exciting things about household material hardship in Gen Peds, I think, is the benefits counseling that is shown using things that exist in the government, SNAP, food stamps, whatever, can actually improve general child health outcomes. But I feel like it's really hard to track enrollment for those things. But theoretically, it could help with the cost of all these interventions. Do you have any ideas on how we could better understand how well we're using programs that already exist and how we can target them better?
I love that question because low-hanging fruit is a good way to go. So there's was a recent abstract and manuscript about to come out. So focusing on one, we're talking here about government means-tested benefits program. So an example of that would be SNAP, or the Supplemental Nutrition Assistance Program, also known as food stamps for food insecure families. Recent data from a pediatric oncology ALL trial show that among SNAP eligible families, just about half of them are successfully participating in SNAP. That's true at the time of diagnosis. And guess what? It's true 6 months into therapy. That means despite our robust psychosocial supportive care resources, meeting with our fantastic, amazing social workers and resource specialists, we're not moving that needle. So that is to say, you are right. We are underutilizing these existing funding resources and we need to up that. Puju Umaratia is working on an intervention to systematically identify benefits eligibility and then do assisted enrollment for families. And I think if that intervention is successful in being well refined so that it can be scaled and centrally delivered, that could be one opportunity, but I think there's a lot of work to be done in this space.
So there's another question online that says, how can you justify doing the support for children with cancer and not doing it for other families without cancer?
I love that question. And I'm acknowledging we're also over time, so I want to be cognizant of that. Please feel free to leave. I will answer that by saying, and oftentimes that question is, how can you justify doing the support for randomized children with cancer and not give it to others ethically? And I have two answers to that. So the first is, I think childhood cancer can be used as a demonstration population to see if it improves outcomes. And while I love the interventions my team is developing, again, I think they're going to fail the first couple of times. And I think until we figure out the right intervention to improve outcomes, you don't then roll that out. But my long-term vision is if we figure out a health equity intervention that works in childhood cancer, that we can justify and fund, because let's be honest, some things are easier to do in children with cancer than in general pediatrics, then the long-term goal would be you take that intervention and first you extrapolate it across all pediatric subspecialties, again, where the argument is a little bit easier. And then eventually you do policy style interventions and you roll that out across general pediatrics. And I think about us using our subspecialty pediatric oncology space as an incubator to develop, iteratively refine, use our clinical trials infrastructure to figure out what works. And then yes, the goal would be we apply that across Gen Peds.
I'll ask. Have you noticed that if biological sex is an important factor in the response to poverty and cancer? And then my second question is, are you aware of potential in vivo models that we could use in the lab space to mimic, say, environmental enrichment to maybe address some of these underlying risk factors?
I love those questions. To the sex by poverty interaction, I don't know the answer. The reality is the translational collaborations I'm doing right now, we're very early in. We have just not had enough samples to look at anything other than the basic exposure and whatever outcome. But I think that'll be very interesting moving forward. With regards to thinking about lab-based models, so are you talking about like PDX models of mice with ALL and?
Yeah. So, giving these rats like gigantic.
Yeah.
Cages to play in and then socially isolating other
Yes.
rodents and looking at the response to say like drug seeking behavior.
Yes. I love that question. And if you know the answer to this, please let me know and email me after. I have not yet been able to find collaborators or protocols that have been well published that fully replicate some of the social deprivation we're talking about. There's a published limited nesting and bedding model in mice, not in anything to do with cancer, that has been used to replicate, for example, housing instability and food insecurity, like that concept of lack of access to resources. But I don't yet know if any of that has been moved into the cancer space. I think it's a very interesting question though, and should be done.
Yeah, that actually kind of leads nicely into my question and follow up to that is there are studies in breast cancer where they've isolated mice and rats and, you know, given them stress because of course they like to be, they're social animals. So if you isolate them, their levels of cortisol increase. And my question was, are you, maybe I missed it, but are you writing into the trials? I mean, it'll be hard because they'll be on glucocorticoids for when they're having chemo and all that stuff, but any evaluation of stress hormone
biomarkers.
levels and also for the nutrition, like to see how much of a factor that toxic stress is, or nutrition, like to look at metabolites in the blood and steroid hormones.
I love that question. Yes to all, it should be done. I have not successfully written in to do all of it. In the trial embedded randomized health equity interventions, we do have included stress biomarker exploratory collection in both parents and children, with the major catch being that there is no validated stress biomarker panel that exists. And so for that, we're using what best has been published. We are getting biospecimens to be able to look at metabolism kind of longitudinally across kids who are enrolled in the intervention or not. But I think there's huge opportunity for all of that. And I have no nutrition data on anybody.
Okay, thanks, that's great work.
Thank you guys so much for your time. I really appreciate it. The nice, all of them.
If there's any students that are gonna have or meet with Dr. Bona after, if you could come down